So thank you for the invite. This is one of my favorite things to do when I have spare time, is play with LLMs and see what I can get them to do. My son and I were coding a Texas Hold'em game the other day. It was a lot of fun. And vibe coding, because I don't code. So I'm going to talk to you a little bit about some of the work that we've been doing on using LLMs, mostly to interpret gene expression. We've also looked at clinical pathology and things along those lines. So the same rules apply. Oh, I am getting a phone call. All right. Apologies. So let's just quick get into this. Get here. So this started for those of you who are in the ESAR group, you're familiar with the TPOD work that was put forth by Rusty Thomas some years ago. The idea being, instead of using traditional pathological manifestations as metrics of adversity, or metrics of potency of toxicological effect, we would look at transcriptomics and short-term studies, collectively put the data together and derive a transcriptional point of departure. From there, you get your reference dose and human exposure. And so I don't want to go too deep into this, other than to say that it's a means of using genomics to identify what equates to toxicological potency. What you get from those studies are a bunch of responses of, in our case, Go terms, right? With the most potent one being the one on the left there. We have these what we call accumulation plots to show basically the firing sequence of biological processes that happen in different tissues, and we declare a TPOD. And for those of you familiar with the ETAP, that's pretty much the only thing you talk about. So first rule of genomic dose response clubs, we don't talk about what happens in this box, so everything up here. But of course, as scientists, this is the thing that we are very interested in. And of course, if you work in pharma, you deal in this space where you have to actually interpret this sort of toxicologically and make sense of it to determine various different characteristics of toxicity that are obviously important in making decisions. What we've historically done in gene expression analysis is we hand it off to our interpreters, right? And all of our interpreters have a certain lens they look through. We have molecular biologists, pathologists, neuroscientists, and they have different ways and what different biology that they focus on and different tools that they use. So there is a sort of this lack of uniformity in process in evaluating gene expression. So the idea of using LLMs as sort of a generalized interpretation tool and to incorporate sort of a more uniform framework in interpretation was appealing, because it sort of helps build some of that consensus. So that was sort of where we're going with this. So this is built off of, when we moving into the LLM space, we have a bigger project we call ToxPipe, which is basically a large language model ecosystem for tox that we are building within DTT. And my collaborators in this space, Trey Sadler, who's since left, are shown on the left here, David Reif, who is the branch chief of the predictive toxicology branch, and Parker Combs, who's now taken over responsibility for the nuts and bolts of this system. And this system there's a chat component, which is based on a technology called LibreChat, and then a number of APIs through Azure to access various different models. So we have access to pretty much every large language model that's out there right now, that we can go ahead and evaluate their capability for various different tasks. So this is, going back to the image I showed you 2 slides ago. The idea being, as you can clearly see, I play around with the image generation too with the AI, is to be able to generate a collection of interpreters and then basically pass the data into those interpreters and then synthesize it into using basically another LLM, and then hand that off to the toxicologist for review, right? So that was conceptually what we put together. And so really what I'm here to talk to you about today is how we formulated the prompt to basically take in the gene expression and interpret it. So I will point out that there are a number of efforts underway in this space already. This is just a point of reference. I'm not going to belabor this. I'll share the slides with everyone if they would like to take a look at these other manuscripts that have been barking up this tree. So the art of the prompt. Yes, there's a little bit of a joke in there. What would the model need to know to create an optimal interpretation? So if you stand back and think about it, the models, I'm sort of channeling for those of you who have started taking these courses in Coursera, Jules White from Vanderbilt. You really have to think of an LLM as like your intern, right? So, and an intern, of course, has some, obviously, a good bit of base knowledge in toxicology. And so when you think about that, what does an intern need to know to help you turn around a interpretation? So what is, and so you have to think about what's in your head as a toxicologist when you're interpreting data, right? It's just think about it from the standpoint of looking at if you had a manifestation in thyroid, right? Thyroid in rats is obviously very responsive to effects in liver because of the thyroid hormone metabolism. So those sorts of pieces of information, like it's important to know that it was a rat that the gene expression came from. So you'd be able to make those connections, but you have to be able to tell the intern that because the intern may not know that, right? Or they may not know. So it's basically an orientation. It's a contextualization process. So one of the first things you do when you're building a prompt is you orient towards what expertise it's supposed to have. So mine, in the case of the gene expression interpretation, you want to have some expertise in toxicological pathology, pathogenesis. You want to be a molecular you want obviously have some expertise in molecular toxicology, in vivo toxicology, and in the context of translating this into something of decisionable value. Obviously, there would have to be some sense of a regulatory scientist or some sort of standards for characterization. So that hence that sort of that additional orientation. Then, of course, you need to know the species, those study details, the species, the strain, the sex that you use, the tissues that you're evaluating, things along those lines. The data, you want to tell it what layout you want, what the layout is, the data that you're feeding it. So we basically have a whole bunch of gene expression. There's a number of variables that go along with it. They're individual genes, so it's fold change, BMD, and then we also have a corresponding rank that we apply to the gene expression. Then you need to give it the goal of the analysis. What do you want to know from the gene expression? There's things like mechanism. How does it relate to toxicity, if it has any knowledge of that? Relationship to apical observations that you may have in the study, or alternatively, observations that you have from additional studies. So say you're running a 5-day study and you have results from a 2-year study. And then finally, and there are more variables on this, and I'm sure many of you online have looked at some of these have taken some of these prompt engineering courses, and there's probably additional variables that you would add here. But then there's things like, the last thing would be constraints, layout, and formatting. So how do you want the response? What's the sequence of information that you would like the prompt to put out? Would you like it to be in bullet point? Would you like it to be in paragraph form? Those sorts of things. And so, these are all things that you have to think about. And I'm going to take you through the details of what we did for the gene expression interpretation. So, and one last thing I would point out that I've found really effective is asking the LLM to help you outline a sort of a detailed prompt once you have the baseline information. And you can ask it to basically continue to ask you questions to go through and fill out the information necessary. And once you get to the end of the conversation, have it formulate or write out the prompt for you. So the first thing we define is the role. So mine is, the role of the LLM is to be a senior toxicogenomics expert, along with expertise in molecular toxicology, veterinary pathology, and also a strategic advisor. They're supposed to have over 20 years experience interpreting gene expression data. They have the responsibility is to synthesize in vivo transcriptomic data with traditional toxicology endpoints for human relevant risk assessment. And then the expertise is in quantitative transcriptomics, including BMD modeling for points of departure and characterizing toxicogenomic hazards. So many of you would probably think of additional things that you might want to add here or modify what I've said here. So, but it's just defining its role so it can sort of orient to the context that context that it needs to have in order to interpret the data. And then the all-important study details, right? So I tend to avoid giving it chemical because I don't want it to be biased, because there's going to be existing information for a number of the chemicals that I have available. But that is an option. What species, what strain, what sex, age at study start, the route, means of administration, vehicle. You get the basic idea. And then there's additional components that we add in, like clinical observations, clinical chemistry findings from the study, hematology. If you're looking at relationships with points of departure, anticipated human exposure, those sorts of, so you can go into ExpoCast and identify, predicted human exposure levels that can then be compared to what your TPOD findings are from the analysis. So as you can see here, there's probably additional features or additional pieces of information that you would probably suggest we incorporate in here, but this was sort of the baseline that we started using in order to just get this off the ground and sort of provide that study detail context. Then the data. So basically the data is going to be fed into the LLM in a structured format and basically incorporated into the prompt. Now, there are ways of using AI agents to go out and basically use tools to take in CSV files and things along those lines. We haven't gone down that path yet. We're simply feeding the information into the model directly through the prompt. And so this basically what this is it breaks the gene list down into 2 lists, up and down regulated, and then it provides basically 3 pieces of information: the genes for each gene, the gene symbol, the potency rank, and the BMD value. And so that's sort of how it's basically every gene is fed in that way, and it's broken up into 2 lists. And so that's just, again, it's just packaged into the prompt, and you can clearly see it's a sort of a structured representation of the data. So it understands basically what it's pulling in. And then next is the goal of the analysis. Interpret the gene list to construct dose-ordered biological events, identify primary mechanisms of action, speculative hazard conclusions related to mechanism and gene expression. Now, this is actually written in much more detail in the actual prompt. I had to pull in specific bullet points here to make it fit on the slide. Justify pathway engagement using potency rank and BMD values. Estimate a transcriptional point of departure based on concerted biological coherent responses in specific biological processes, and deliver a cohesive data-grounded narrative stating necessary assumptions. And then finally, we have another component, which is, I believe this is the last one, the uncertainties. Acknowledge key assumptions and data gaps explicitly. Indicate likely direction and magnitude of bias for each limitation. Reconcile transcriptomic signals with apical endpoints and timing. Distinguish adaptive pharmacology from adverse if you can, and conclude with confidence rating and top follow-up actions. And then finally, this is just the constraints. Tells you how many words you want. It is in paragraph form, bullet form, however you prefer your output. So we built a tool, and this is Parker's work, to basically be able to load in data from our tool called BMD Express, which does the genomic dose response analysis. This is focused on interpreting, individual genes, as I pointed out earlier. You load it up, and then you apply. You basically identify which columns. So it's a big spreadsheet. You identify which columns you want to pull in to provide the information about basically each individual gene that you saw on the data set, the data page there, or the data slide I showed you. From there, you formulate your prompts. So once the data is loaded, so this is just an example of our prompt that we've been using. We've been iterating quite a bit on that. And then you also have the input of all of the, basically all the study parameters. So species, tissue, things along those lines. We've been iterating on that also. So basically, this is what the data look like when we get it in there. This happens to be from a gene expression study in liver using Furan. So individual genes on the left there. The ranking based upon the potency. The ranking is provided just to allow the LLM to more effectively identify the sequence of firing of the genes in the study. The fold change, or the BMD value, the fold change, and the direction adjusted fold change. So basically, the direction adjusted fold change comes from the overall direction, and it's just an output from the BMD Express software. So it can understand whether the gene's going up or down, and that will facilitate the interpretation. So I don't want to take too much time here going into too much depth on this. I have documents that I'm happy to share with everyone with the prompts I used and the outputs and things along those lines. Again, this was more oriented towards the concepts behind prompt engineering and actually getting the data into the LLM and orienting it so it can actually give you an answer. But just a quick overview, we ran three models to be able to then bring them together into a consensus. So we ran Claude Opus 4.1, GPT-5, and Gemini 2.5 Pro, which those three are sort of the top level models that are sort of available now. And then finally, we ran GPT-4o to basically, we fed all of the results from each of the three that were three that I showed you just before there into GPT-4o, and then it came up with the consensus characterization. And one of the things that we were that you struggle with LLMs is this issue of consistency, because basically you can feed the same information into the same LLM multiple times and get slightly different outcomes. And so one of the ways that we've been trying to deal with this and adapt to the issues associated with some of the hallucination and things along those lines is looking at three different models and then using that information to then formulate a consensus output. And so just to summarize I'm not going to go through the three models, the three main models that we looked at, but I'll talk about the consensus here. So all three models agreed on the primary hepatotoxicity mechanism involving oxidative stress, progressing to mitochondrial dysfunction, cell cycle dysregulation, and hepatocellular injury. The transcriptomic pointer departures range from about 0.12 to 0.7 mg per kg per day. This is consistent with what we derive with our standard approaches using BMD Express. GPT-5 did have a slightly different TPOD of 1 mg per kg per day, so slightly higher. The dose ordered mechanisms, they showed phased progression of effects. Lower doses showed xenobiotic metabolizing enzyme effects, early stress responses. Intermediate doses, we saw intensified cell cycle checkpoint activation, DNA damage, mitochondrial dysfunction. And this subsequently, at higher doses, this led to signals of apoptosis and hepatocellular injury. There was clinical pathology in these studies that we could incorporate into this, so that was another piece of information. And it found that elevated ALT and AST levels are consistently observed, indicating apical hepatotoxicity with high confidence in this clinical indicator. So you can see how it would link back at 5 mg per kg today at higher doses to apoptosis and the cellular injury that was seen in the gene expression. And then there's a collective risk implication. We didn't give it human exposure. So obviously you can't do a margin of exposure. So Claude and Gemini advised human exposures well below 0.5 mg per kg per day. GPT-5 suggested 1 mg per kg per day. Obviously, with safety factors, those are going to be much lower. And then all models acknowledged uncertainties, including lack of toxicokinetic data. So it was aware that we didn't provide it that. Histopathology, which we didn't have available. Species differences, which is also a concern with when you're evaluating non-human studies. And then moderate confidence in risk recommendations and high confidence in identifying limitations. And so what you get is this is what this here what you're seeing here is a small fraction of what it actually puts out. I couldn't fit everything on the slide to give you the full breakdown and detail of what it did, particularly for the dose ordered mechanistic characterization, and I will share that with the group. But again, this is more about the building of the prompt in order to get the data interpretation to occur. And so what I was hoping to do is provide you enough of a sort of frustrating representation of this process, because it's basically a somewhat of a art form at this point, that you would engage and think through how you would engineer a prompt to take in gene expression or clinical pathology in order to provide a quality output from these models. Because ultimately, I imagine that the field is going to evolve into this space where it's using these models, just particularly for things like clinical pathology and gene expression, that is going to become sort of the standard. So on the front end, we're going to need to really get a handle on how, what the standard should be for writing the prompts for, and then for evaluating the outputs. So I don't want to go any further, and I want to give other people a chance to present. So I'm going to close it off there because I want to have some discussion at the end, and I want to nurture that interaction.